The number of students is so less. Okay, so we'll start this class. Do you all remember what we have studied 2 days ago? Not 2 days, I mean in our last classes. ELISA, direct and indirect ELISA. Do you remember now?
Yes, ma'am.
Yes,
Yes,
ma'am.
ma'am.
Okay. So we there is another ELISA type of ELISA which is known as sandwich ELISA. So we'll study on that today. Okay. I'll share the screen with you. Just so this is direct and indirect ELISA, which I had explained in the previous class. The another ELISA which remains to be studied about is sandwich ELISA. Now, when we say sandwich, what do you understand by that term? Sandwich. Sandwich means in between something, in between two things. Okay, so that is sandwich. So the same reason, for the same reason, the word sandwich has been used in this particular technology. So why is the sandwich ELISA used for? It is used for detection of antigens. Then it is also used for home test kits like pregnancy test kits. In such kind of tests, the sandwich ELISA method is used. So here the word sandwich has been used. As I told you, sandwich means what? In between two things. So here those two things are different antibodies. And the thing which will be in between both the antibodies are the antigen, the target antigen. Okay, so that is the reason why the term sandwich has been used. So these antibodies, they bind to different epitopes of this target antigen. There will be one antigen only, and two different antibodies will bind to this to the epitopes of this antigen. So that is why this particular test is known as sandwich ELISA. So we learned how the sandwich ELISA is performed. To perform the sandwich ELISA, as I told you, we require two antibodies and one antigen. So here you see the antigen that will be provided first is known as the capture antigen, capture antibody. Sorry, this is the capture antibody. Then after that, in between will be the antigen, the target antigen, and then on top of it, the another antibody will be added, and that antibody will have the enzyme linked to it. So that will be the enzyme linked antibody. So these three elements are the necessary things that are required to complete the sandwich ELISA. So how do we start first? So we are taking a microtiter well. That is the first step. In this microtiter well, what are we going to do? We are going to add some antibodies. So first of all, the antibodies we add are the capture antibodies. So capture antibodies that will be specific for that particular antigen will be first of all added to the microtiter well, and then allow those antibodies to immobilize so that they do not go away from there. After this antibodies have been added and then they are immobilized in the microtiter well, next thing done is the well is rinsed so that the unbound antibodies are removed. After the unbound antibodies are removed, the next thing we need to do is add the serum sample. The serum sample will have the antigen in it. So that and the serum sample containing the antigen will be added to the well where the capture antibody is present. Now that serum sample, you see when the serum sample was added, there are several different types of structures you see are present in front of you. Now what are the structures? These are antigens. Now the antigens that might be present in the serum sample is not necessarily has to be only one type. There may be several different types of antigen present in that serum sample. But the one which is specific for this antibody will only go and bind to it. Everyone cannot bind because this antibody is specific to one particular antigen. And we do not know which antigen out of this will be specific for this. So the one which is specific will go and bind to it. Okay. So here we see that on both the sides of this antibody, the antigens has bound. After this, again, the next step will be followed. That is the rinsing of the well to remove the unbound and non-specific antigen. And then the third step is performed. That is addition of the enzyme link antibodies to the well. So these enzyme link antibodies will again go and bind to these antigens, the epitopes of the antigen. So here we see that the antigen is in between, and on top of it the enzyme link antibodies, and below it is the capture antibodies. And then the extra antibodies are again washed away by rinsing the well. After this, the substrate is added to the well, and then this enzyme and substrate, as they are specific to each other, the color change will occur, and then the antigen will be identified. This color development indicates that the positive reaction has taken place. So that is how a sandwich ELISA is performed. Okay. Now in this sandwich ELISA, we find two different types of ELISA again. So two different sandwich ELISA. One is direct sandwich ELISA, and another is indirect sandwich ELISA. This is similar to direct and indirect ELISA itself. So, but the procedure that they follow is that the antigen has to be in between two antibodies. So here you see, just now what we studied was direct sandwich ELISA, and had it been indirect sandwich ELISA, the capture antibody would be there, the antigen will be added, then the primary antibodies will be added to it, and then the secondary antibody, which has the enzyme attached to it, will be added. So that is how an indirect sandwich ELISA is done. It has got capture antibodies, primary antibodies, and secondary enzyme link antibodies. Okay, so these enzyme link antibodies are common in both the cases. That is required because we add the substrate for the change in color. If these enzyme link antibodies are not there, then the color change will not happen because of that. Okay, so this is how these two different types of ELISA are performed. Okay, so that is all about sandwich ELISA. Did you all understand it?
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
Okay. Another technique that we have to study is known as RIA. Okay, so what is RIA? Okay, so this is radioimmunoassay or RIA. So what is this? The technique in which a radioisotope is used as a tag or label for the detection of antigen antibody complex. That is, a radioisotope is covalently linked to antigen or antibody. So this is known as RIA. So what happens here? In this assay or technique, an isotope, a radioisotope, is used to label an antigen. That means it is added to an antigen. Just the way you saw in our previous techniques that enzyme was labeled to an antibody. Similarly here, what is that? A radioisotope is labeled to an antigen or antibody. It depends on what you want to look for. So depending on that, that radioisotope is labeled to it. So those kind of antigen will be known as radio-labeled antigen, or if it's an antibody, it's known as radio-labeled antibody. Now this is a history about this particular technique. This technique was developed in the year 1959. It was developed by two endocrinologists, Rosalind Yellow and Solomon Bersen. Then in the year 1977, Rosalind Yellow was awarded Nobel Prize for Medicine. But Solomon Bersen was also he also had to he also had a share of that Nobel Prize, but because of his untimely death, he could not receive it. He was already dead. So it was only Rosalind Yellow who was awarded this Nobel Prize for medicine. Okay, now coming to the radioisotope. Now see here, you can see this is an antibody, and on this antibody, this isotope has been labeled. Okay, so these radioisotopes are covalently attached to antigens or antibodies, and they are, since they are radioisotopes, so they can emit radiation. Okay, so these radiations which they emit, that can be measured. That can be measured by instruments such as gamma counter. That is the name of the instrument that is used for measuring the radiations that is emitted by this isotope. So if I give an example of a radioisotope that is most commonly used in this kind of studies, it is iodine 125. Iodine 125 is a radioisotope. Now, what is the basic principle behind this particular assay? Radioimmunoassay determines the concentration of an antigen in a sample based on what? Okay, the prime principle of performing this assay is that it helps us in determining the concentration of antigen that is in a sample. That means you have a sample of human patient sample with you, and then the person has got some antigen in that particular sample. So you want to find out the amount of antigen present in that particular sample. For that, you need to perform this radioimmunoassay. Competitive binding between radio-labeled and unlabeled antigen for its specific high affinity antibody. So there is to perform this technique to find out what is the concentration of antigen present in the sample. Well, there we perform a competition of binding between the label and unlabeled antigen to find out its specificity for the high affinity antibody. So you probably may not have understood what I have said just now. You will understand. We'll move on. That will help you to understand. So you see this technique. This is a microtiter well that have been taken. In this well, they have added antibodies. Okay, they have added antibodies, and then they are now next going to add antigens to it. Now these antigens have, why are this, what are these yellow colors that are present on this antigen? This antigen, that means, are radio label. And here when they have written antigen here, see here they have written antigen, antibody, which is designated as AB. Antigen is designated as AG, and this AG has got a star mark here, an asterisk mark here. So this asterisk mark denotes that this antigen is radio-labeled. Okay, so you can see this yellow color thing on top of this antigen, this red color antigen, this yellow color thing here means that they are radio-labeled antigen. So this radio-labeled antigen has now been added to the well, and then they have, since they're specific to this antibodies, they went and found itself to these antibodies. The one which could not bind to those antibodies have been washed away. Okay, so here in this particular well, if we try to find out the radioactivity, how much radioactivity or how much radiation is it emitting, if we try to find out, we'll find that it is radiating 100% of radioactivity. The reason is because all are radio-labeled antigen. They all are radiating. So that is the reason they are 100% radioactive. Okay. Now we'll take another well, and again we have added antibodies to it. Antibodies to those well, they have been immobilized here. Now we will be adding test sample. We will be adding test sample, that means serum sample here. Now that serum sample will consist of antigen. We will not add, we cannot tag those antigen with this isotope, radioisotope. So those antigens in the test sample will have no radioisotope, and another antigen will be taken which has radio labeled with it. So both of them will be now added to this microtiter well. The ones which are radio labeled, another ones are the antigen, just a test sample which has the presence of the same antigen specific to those antibody. Those two have been added. Now what will happen, there will be a competition between these two, both the antigens. Because you see here, the antigens, whether they are radiolabel or not radiolabel, antigens are same. It is just to separate it from them. We have radiolabeled it so that we can distinguish that which are radiolabel and which are not. We are to perform this test. Actually, we have radiolabeled it, but the antigens are same. Both the antigens are same. So obviously, both the antigens will be specific to this antibody, and that is why this time not necessarily all the radiolabeled antigen will go and bind to the antibody. It may be the non-labeled one also. Anyone can go and buy. We have washed away again to remove the unbound ones, and this time we again checking the radioactivity of this. Now we find that the radioactivity has decreased compared to the previous one because this time all of them are not radiolabeled. 50% are radiolabeled, 50% are not radiolabeled. That's what we see in this picture. So here the radioactivity has decreased. So decrease in radioactivity tells that the antigen is present in the test sample. So this has helped us in proving or in finding out that the antigen is present in the test sample. Previously we did not know. Obviously, by just taking a sample, you will not know whether the same antigen is there or not in that sample. You will have to perform the experiment. And after performing this experiment, this has been proved that the same antigen were present in a test sample. Now, what we next need to do is find the concentration of the antigen in the patient's serum. How much amount of antigen is present in the serum? We have found out whether that antigen is present or not. That we got to know. Now we need to find out what is the amount. So to detect the presence and quantity of an antigen in the patient's serum by using radioimmunoassay. What will we have to do for that? To find out to find out the quantity of the antigen in the serum. So to find out the amount of antigen in the serum, we will have to obtain a standard graph. So how is the standard curve prepared? It is generated by measuring the amount of radioactivity. How is the radioactivity measured? Using fixed concentration of the radiolabeled antigen. Fixed but excess concentration. That means the amount of radiolabeled antigen has to be fixed. That means you decide that I'm going to take 10 radiolabeled antigen, but it has to be in excess amount. The amount has to be more, excess concentration. Then varying known concentration of unlabeled antigen. And this time, the unlabeled antigen you take has to vary. Means different concentration, but it has to be known concentration. For example, you take different like 10, 20, 30. These are different numbers, so different concentration, but known. That means you know it that you are going to take 10 amount of antigens, 10 numbers of antigens. Then in the next well, you're going to take 20 numbers of antigens. So these are varying but known concentration. Okay, so these are the two things you need to take in order to measure the amount of radioactivity and then to generate the standard curve. Okay, so then what are the steps involved in the generation of the standard curve? Now what we need to do, what are the steps that are to be followed? So first of all, what they have taken? They have taken a well, microtiter well, and in this microtiter well, they have this antibody in fixed concentration. The concentration of this antibody is fixed. That is 3 numbers of antibodies they've taken and they've immobilized in this well. Okay, now they are taking, they've taken this antigen which are radiolabeled. Here we see with this asterisk marks in this antigen, and here the amount of this what do you say, radiolabeled antigen is fixed. That means 8 numbers of radiolabeled antigen they have taken, and it is in excess concentration. In compared to the antibodies they've taken, the amount is more here. Okay, so in excess concentration, the radiolabeled antigen have been taken, and now they will be added to this microtiter well. After adding to the microtiter well, okay, just see this experiment. It's a continuation of the previous one to make it more easier for you all. Now see, This is the well that they have taken. As I told you, they have taken 3 antibodies. Okay, so this is a well. This is, I mean, this is a slide where they have taken five different wells. Yeah, in each well they have used 3 antibodies. The number of antibodies is fixed, 3 in all of them. Okay, now what do we do first? First of all, amount of radiolabel antigen is added in the first well. Only in this first well, the fixed amount of radiolabel antigen is added. So 8 numbers of radiolabel antigen has been taken, and then they have been added to this first well. Okay, so you see all the antibodies uh, have bound to this radio-labeled antigens. Okay, and then the unbound antigens are washed away. Then after this, the radioactivity of this particular well is measured, and it is found that 100% radioactivity is happening here because all the radio isotope are bound to the antibodies. So this is 100%. Okay. Then rest of the well, what they will do, to all the other remaining wells, we will add same fixed amount of antigen. That is 8 numbers of antigens were used in the previous one. So here also they are adding 8 numbers of antigen, but increasing known concentration of antigen. That means the concentration of antigen that they are going to use in a test sample is uh, going to vary in all of them, increasing. Here if they take 4, here they will take 8, here they will take 12, this they will take 16 here. So it will increase in concentration. Now, after addition, the antigen antibody will compete for antigen binding sites in the well. Since both the antigens are same to each other, they will all compete with each other. So here, the amount of unlabeled ones are less compared to the labeled ones. Here it is slightly more, this slightly more. Here the unlabeled ones are in the most amount, the highest amount. So those antigen which are labeled and which are unlabeled, both will try to bind to this antigen binding sites of the antibodies. Okay, so they are increasing in concentration. Now they will again be washed away, rinsed, and unbound ones are washed away. So what do we see now? That radioactivity of all of this well will be next measured. Here in the first well was measured, it was found to be 100%. Now in this remaining wells, the radioactivity will decrease. This had highest radioactivity. This has gradually lower. Then this will have the lowest radioactivity because the amount of labeled antigens are less compared to the unlabeled ones. So that is the reason they have the lowest radioactivity. Okay. Now, after this, we need to prepare the standard curve. Now, how this standard curve is prepared? The radioactivity percentage is kept on the y-axis, and concentration of antigen is kept in the x-axis. As the concentration of unlabeled antigen increases, the radioactivity will decrease. Remember this. The concentration of unlabeled antigen will increase. That means when this will increase, concentration of unlabeled will increase, the radioactivity will decrease. It will, so this curve is starting from top. It's not starting from down here, not from 0. It will start from top. So depending on the amount of radioactivity, if it's it will go on decreasing. So this is how the standard curve for this particular test is formed. Now we need to find the quantity of the antigen in the patient's serum, in the serum of the patient. How much antigen is present? We need to find out that. So we are going to follow the same technique again. The microtiter well was taken. The antibody has been added to it. It has been immobilized, and antigen which are labeled are added to it. Okay. After addition to it, we also take antigens. We also taking now the patient serum in it. Okay, the patient serum will have the presence of the antigen in it, and then they will both be added, and there will be competitive binding in between the two. The unbound ones will be washed away. So here we see that both radio labeled and unlabeled ones are added. Okay, now the radioactivity is measured. When this radioactivity is measured, we don't find it to be 100%. Yeah, it may be as much as 35%. It depends on how many radio label has bound to the antigen with the antibodies. Then we, like the standard curve which was formed, we plot the graphs and then we find the concentration of the antigen on it. From this, we find out the amount of antigen that are present in the serum. So this particular test is mainly used to detect hormones and detect allergies. Then the demerit of using is that the person who work on this, they are exposed to radioactivity, and so that is a bit hazardous for those persons. And most of the radioisotopes have got short shelf life, and the instruments that are used for this technique are very expensive. It's a time consuming method. Okay, so is it clear to you all?
Yes.
Yes, ma'am.
Don't have any doubt?
Yes, ma'am. No, ma'am.
No, ma'am.
Okay. Okay then. We'll stop here. I'll take your attendance now. Suman,
Yes, ma'am.
Tushar, Mona Lisa, Kanan, Bitukon, Deepjyoti,
Present, ma'am.
Nikita, Nilpavan,
Yes, ma'am.
Sayasri, Hirok, Samir, Irani, Barkha,
Present, ma'am.
Barkha, Suparna, Anjan,
Yes, ma'am.
Amit,
Yes, ma'am.
Shreyashi,
Yes, ma'am.
David, Alina,
Present, ma'am.
Anjali, Sania, Jintumani,
Yes, ma'am.
Sanchita,
Present, ma'am.
Suman, Darshana, Swapna,
Present, ma'am.
oh, this is Swapna,
Yes, ma'am.
Yaljomo, Indira,
Present, ma'am.
Shivaji, Moromi,
Present, ma'am.
Rajashree,
Present, ma'am.
Ritumani. Is there anyone who could not respond because of the internet connection? You can write your roll number in the chat box if you're facing trouble. I hope everybody's marked, right? You can just go through once. I hope no one's left out. Okay. So I'm updating this attendance.
Exam